This is episode 75 of the Immunology Podcast, Autoinflammatory Diseases with Dr. Scott Canna. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Scott Canna from the Children's Hospital of Philadelphia on the podcast to talk about his research on auto and hyperinflammation. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
we can't wait to attend Immunology 2024 in Chicago. The meeting takes place from May 3rd to 7th. And we'd like to remind everyone to make sure you've registered and that you have booked your hotel. Learn more at immunology2024.aai.org. And not only that, because we are also very excited to announce, Jason, right, that this year's program will include the first ever live show of the podcast, the Immunology Podcast, going live at AAI, taking place on Saturday, May 4th from 12:30 to 2 p.m. Jason and I will be hosting a live recording of the podcast focused on how AI is being used in immunology research. We hope to see you there.
Oh, AI at AAI.
How exciting. Oh my God, a live show. This is, I'm just so excited.
Well, and in a year right after this one, it's in Hawaii, so we can do it live from the beaches of Waikiki.
That has to be our goal. First things first, Chicago. It's like in 6 weeks or something. Super exciting.
Look, I'm not thinking about that. It's spring break time. I'm not thinking 6 weeks from now.
Yeah. So what is your spring break plans?
Vamos a la México.
La México, no, El México. Oh, México. Well, enjoy your warmth because you're definitely not getting that warmth here in the north of Europe. So enough. I don't want to hear more how much fun you're going to have in Mexico because
you'll hear about it all when I get back. I'll have some stories.
Oh, Christ. So why don't you start up with the science?
I have two related stories today. So you're going to hear about microbial metabolites from me now and later, but I'll let you pick, Brenda. Do you want to hear about microbial metabolites in infection or microbial metabolites in cancer first? You'll get the other one later. Cancer or infectious disease?
Oh man, let's go for infection. I think cancer always get the spotlight.
Yeah, we got to respect infectious disease. I appreciate that from you.
Indeed.
All right, so one type of bad E. coli is enterohemorrhagic E. coli or EHEC. Known by serotype 0157H7, bloody diarrhea, very bad news, right? When people eat bad burgers and get E. coli, this is what we think of. So we know that the host microbiome, right, that microbial milieu in your gut wages war with all these pathogens all the time and holds things in check. But how it does this in all specific cases is relatively unknown. So this paper tries to solve this again in mice models, because it's not ethically to do some of this in people, right? But looks at Citrobacter rodentium, which is a mouse AE, which is attaching and effacing pathogen, of which EHEC is another type. But if you think about what you'd want to give a mice, you're going to give them Citrobacter as the model organism. And so they are trying to understand how the host microbiome could influence all of this. So there's ideas of the host microbiome primes immune cells to kill things. It competes with the space. That's all true. But this group found something else super interesting, which is tryptophan metabolism and altering actin pedestals through the dopamine receptor. So what did they find? So they found that dietary tryptophan protects against infections with this strain. Okay. Well, dietary tryptophan is one of those things that's very well known to be metabolized into a lot of downstream indols and other things. I'm not going to go in all of them, but it's well known to be a very the bacteria really like chewing it up, spitting it out, and doing things and making metabolites that have important biological function, which is why it's one of the things you go after. So they showed first off that the tryptophan itself, it wasn't altering the microbial milieu. Right? So it wasn't altering the microbial milieu. It wasn't changing the bugs in your gut. Neither were the metabolites. But tryptophan and the metabolites, the bacterial metabolites that you can just supplement, affected the ability of the bacteria to bind, Citrobacter dedentium to bind and cause pathogenicity. So that's saying that it's something about the metabolites itself from the bacteria. If they did germ-free or antibiotic studies, got rid of the effect, right? So this is very, so they did antibiotics as the main system here. And if you antibiotically ablate the mice and then try to do this and give them Citrobacter, they all fall over, right? So it's microbiome mediated. And if you give them tryptophan and antibiotics and then try to do this with the Citrobacter, it also don't work, right? You have to, because they're decolonized with whatever's producing it. So then they looked at what was predicted by these molecules to bind to, and it's dopamine receptors, DRD2, 3, and 4. And they used haloperidol, which is a pan inhibitor, and that reduced the effect of the metabolites. And then they did CRISPR knockout in CACO2 cells, which is a human epithelial intestinal colon cancer cell line, to just see if they could get the signaling to work and knock out one of them. And they found it was DR2. So they found that only one of the receptors was really important for it. And they did some GPR signaling and also showed that it was going through the GPCR with some glow sensor and Tango G protein coupled receptor assays to look at us. And they had say, all right, it's a tryptophan receptor acting through these metabolites, specifically this one. They made some villain-Cree knockout mice, right? So villain-Cree DRD2 knockouts. You knock it out, you lose protection. If you knock it out in myeloid compartment, you don't lose protection. Okay, so that's pretty cool. So it's only the gut cells. And so like, well, how the heck is it doing this? So they do some other staining, and it sees that what's going on is that there's this active regulatory protein called NWASP. It's initial host protein that's hijacked by these pathogens. And so they found that these reduce the pedestal formation. So these metabolites reduce this actin pedestal formulation that's regulated by NWASP. And its effect, then the regulatory effect of that is through the DRD2 receptor. So this regulation and dropping of the pedestals goes away if you knock out the receptor. And so then the general story is dopamine activation by bacterial metabolites drops the level of actin pedestal receptors, which means the bacteria cannot use that to hijack and attach to the cell. But this is a Nature paper, and let me pause here. I forgot to mention at the top of the hour the full name of this thing. I jumped right in. So this is dopamine receptor D2 confers colonization resistance via microbial metabolites. First author is Samantha Scott. Last author is Pamela V. Chang. And it was, this is interesting, received 13 September 21, published March 13th, 24. And it was accepted February 7th, 24.
Well, it's only 3 authors, right? So...
It's only 3 authors. The middle author is Jingjing Fu. But this had a 3-year turnaround time for acceptance.
Well, if you have to do all the revision yourself.
It's an article in Nature. So not one of the big ones. It's one of those short, the famous short Nature papers. And so then they try to figure out how it's working, right? So it's this receptor and it's doing all this. So they try to look at what part of the G protein coupled receptor is, and they isolate this down to its being G beta gamma signaling, not G alpha I or beta arrestin. So it's through that. And the signaling causes degradation of the NWASP pathway. So it's causing NWASP degradation by phosphorylating, leading proteasome degradation. If you lose NWASP, you lose pedestals. So they mapped out almost the whole pathway.
That's quite impressive. I don't always say this almost every single time we discuss one of these microbiome, but it's just so incredible, like how many gears this whole thing has. Yeah, how basically the activation of this the neurotransmitter receptor that you would think is has such a clear effect on the adhesion. This is the adhesion of the pathogen, right?
Yep. Because they bind on and they start stripping off your epithelia, but they got to bind first.
Right. And then if you consume tryptophan, then
other bacteria will take your tryptophan and
Right.
convert it into tryptophan metabolites that bind to the DRD2 receptor, which
Right.
increase the number of pedestals and prevent Citrobacter from binding.
Very interesting.
So
Very
a low
interesting.
tryptophan diet would then make you more susceptible to infection.
Yeah. Well, I was at the symposium today, and we had Caetano Reis Sousa, who was, a guest at the podcast, and he also had this huge story and about how vitamin D also had a protective effect. In this case, against cancer, but I thought it was so good.
One of my most cited reviews is about vitamin D as an immunomodulator that I wrote a while ago.
Oh well, you would like his. I thought of you when he was presenting his research.
Yeah, too much, too little actually leads to both it makes a bad immune system. It's just not like it's hypo or hyperactive. It both can't kill pathogens and attacks you more. It's just bad. It's like more autoimmunity and less killing of things it should be. You don't even get the benefit of hyperactive immune system killing stuff. It just wants to hurt you and not the things it should be going after.
Well, what the things it was one of the pathways that was regulated was indeed cross presentation by CDC1s, and the capacity of these dendritic cells to take necrotic cells and process their antigens for cross presentation. That's super interesting. All right. Well, I also have a pretty good story to follow up. Um, this is a paper from the lab of Diane Mattis. So we will know her well. Very well known for her work on uh, regulatory T cells, and also on one of her fortes is understanding how antigens are presented to T cells inside the thymus that contributes to the training of the T cells in the thymus. She discovered the gene AIR, the transcription factor that regulates the expression of all these pan tissue genes within the thymus so that T cells can see them. And this paper is called AIR, relies on cDNA to flag gene targets for thymic T cell tolerization. First author, Juan Fang, and was published in Nature as well. And this is really, really interesting because so we know that AIR is super critical for the expression of all these antigens in the medullary epithelial cells in the thymus. But it is not clear how AIR works, because AIR doesn't really recognize uh, um, a specific motif so it's not completely clear how it actually does that. So trying to understand this, the authors, they kind of relied on they did two different things, talking about AI and immunology. And the first thing that they did, which I have to admit, it's a little bit hard to understand really all the details, the technicalities. So I'm sorry if any of you machine learning experts, this don't if I didn't get it right, but well, basically they build a neural network. They call it a convolutional neural network, in which they input. So they do it in kind of two phases. On the one hand, they train the network to kind of understand promoter regions. So they just feed it a ton of different promoter regions. And apparently this helps the neural network to understand promoters kind of. And then on a second, what they call the fine tuning stage, they actually feed it like promoters, and they say these are AIR induced or these are AIR neutral genes. So it allows the model to learn how an AIR induced gene looks like. And so they do this your thousand of sequences from the B-Black6 genome. And basically, they reached the conclusion that there's mainly two motifs or two things that stand out that they look further into. And one is CA repeats, and what is would be binding motifs that are bound by NFE2 and MAF, which are also transcription factors in this case, binding together and recognizing certain motifs. And this seems to really stand out as part of the features that affect AIR's target gene specificity. And the repeats are pretty interesting because they are well known to be said DNA forming sequences. And said DNA is this, weird, non-canonical, helical form of DNA that goes like the other way. So it looks pretty weird. Looks like a little bit more. I think I've learned in like biochemistry class. I guess said DNA is just there, doesn't do much. Apparently, that is wrong, and I'm learning it now. So, said DNA is present. There are motifs across the mammalian genome, and it's present and it's formed. And this plays a role in gene regulation, and they are often enriched at gene promoters, and they're possibly correlated with promoter activities. And this seems to relate to the fact that the formation of these said DNA structures generates a chromatin or a structure that is more open, or it's also more has a higher tendency to or more fragile. They have a higher tendency to generate double-stranded breaks. And this is something that double-stranded breaks regulate transcription and oftentimes promote initiation of transcription. So by having set DNA, actually, that kind of poises this genetic sequence to be transcribed. Sorry, and so they. So this is in their mind. This is very interesting. So they look closer into that, and they do find that set DNA seems to be um, preferentially formed in the gene promoters of these epithelial cells in the thymus of mice. And they can see they also check out cDNA. They do some immunoprecipitation where they kind of also seem to identify that there's an enrichment in said DNA in these regions. They also use the treat cells and the treat mice and also cells with spermidine that I didn't know stabilizes said DNA formation. So that's very interesting. And this seems to allow the air to upregulate the expression of these genes at a higher level. So it doesn't really change which genes are being are being promoted or being induced by air, but it does increase their basal expression and in wild type mice. So they make a very convincing, and they do a lot of analysis. I have to say, they make a very convincing point that um, indeed these different structures is part of what air relies on to find the genes it's supposed to be transcribing. So that is basically the main idea. They also look at this. Another thing that they do as part of the experience that they do is they compare different mouse strains and the different kinds of SNPs that they have, and they correlate this with the expression of genes in medullary thymus epithelial cells, and they show and they do all of these correlations with all these really complicated algorithms, and they kind of show that this correlation with DNA and some other transcript, and also in the case of the NFE2 MAF binding motifs, they also show that they really seem to be poising air to go to that particular genomic sequence. So it's in a way guiding where this transcription factor goes to in a more indirect way. So I think it's very interesting because it's really cool for us to understand how is this happening in the thymus,
I think that's an interesting mechanism. Uh,
right?
Oddly evolved to be that way. I have lots of questions on the evolutionary biology of that.
I know, me too. I also thought it was very peculiar. I liked. I mean, I there's a lot of this paper that I don't understand. I'm gonna come clean here, but I thought it was very interesting.
All right. Well, keeping it moving,
Mm-hmm.
next paper in Cell is microbial metabolite enhances immunotherapy efficacy by modulating T cell stemness in pan cancer. It came out March 28th, so in the future. This is, the publication dates sometimes in the future when you get the papers. First author, I'm going to butcher all of these names. My opinion is not fluent enough. Liangjing Jia is the first author, and dual corresponding authors are Shujie Chen and Liangjing Wang. So here you go. We know that there are good microbiomes, and no one can see my air quotes, but you can hear them, and bad microbiomes for immune checkpoint blockades. And then if you put someone who's not responding to checkpoint blockade, and you have either a patient with a good who's responded to it, or you just get some healthy people and try it out, you're going to, depending on if those healthy people have the good microbiome, you can enhance your response to checkpoint blockade inhibitors, especially PD-1 inhibitors. So what's associated with this and somewhat known is that low lactobacillus in the gut is associated with being bad. So they wanted to figure out what was going on here. So they used an MC38 model of colon cancer. And so they used that as the model system because PD-1 works really well against that, right? And so they started with that and then similarly went in with some follow-up models with APC-Min, which is spontaneous colon cancer, or AOMDSS, which is chronic colitis-induced colon cancer. So those are the three starting systems. And what they did is they just took some mice in different colonies and different housings and gave them anti-PD-1 in the system and found responders and non-responder groups. Then they transplanted feces from the non-responder to the responder group to the non-responder and see if they could recapitulate the phenotype, and you could. And then they sequenced them and tried to find the difference. And lactobacillus in the low responder was low, and L. Johnson was the most prevalent of those. L. johnsonii, two I's at the end of the Johnson. So that was an important organism that seemed to be driving the response. So they looked at oral administration and, if you just give them the bug, it also works. So what's happening and how is this working? And so they do a bunch of dead prep. So they cook it, they ultrasound it, they do conditioned media of it or the bug, and they find you have to have the bug or the conditioned media to work. So you can't so something about what the bug is producing in that conditioned media is really important. So they found, they did some comparisons between broths with and without L. johnsonii and found that it produced a lot of, guess what I'm going to say for a metabolite? Tryptophan metabolism.
Oh, come on. It's always tryptophan.
It's tryptophan, also known as osmophan. And if you give them a tryptophan negative diet and then give them L. johnsonii, the protective effects go away. So then what is it? It is IPA, which is an indole, that is known to be a tryptophan metabolite. And they found this in the plasma, but only in mice fed tryptophan. So this IPA goes up in the plasma. And then if they gave IPA, it also helped. Huzzah! It's amazing. So how does it work? And we're not quite done. This is where it gets wild, but we're going to take a detour. Well, what's happening here? Well, it promotes TPX cells. These are a subset of precursor exhausted T cells, which are known to be important for reactivation with PD-1 blockade. And there's more effector cells. And so I've skipped to the end of the paper here. I'll get back to the other crazy part. But they show that how it works is these metabolites induce TPEX cells, activates them by modifying the H3K27S acetylation site of the TCF7 gene at its super enhancer spot. So you open up the super enhancer because of this metabolite. Now, how exactly that's happening, they can't quite figure that out. But they get that far. And that results in more TPEX and more T effector cells, which have clonal expansions and which go and kill the cancer. Okay, great. So L. genocide, we should just give people L-Johnsonii, and that's going to solve all the problems, right? Right? Well, wrong. So,
Man, you got me right there.
I know. So this is where it gets clever. So IPA is the downstream end of the pathway, right? L-Johnsonii makes an intermediate called ILA, and they find a gene that's important in it to make ILA. But then they know about that, but they know it does this, but it doesn't make IPA. Well, but then how does L-Johnsonii, do such magic? Well, they find out that you need C. sporogenes or other organisms, but they hone in on C. sporogenes. And they found that oral gavage of L-Johnsonii increases the level of abundance of C. sporogenes, which is probably how they do the jump down this. And if you do single germ-free association of either bug, guess what? L-Johnsonii doesn't do anything. But if you mix them both together, it does. And so what actually happens is L-Johnsonii makes ILA, and ILA goes and hangs out with C. sporogenes, and that makes the IPA you need for this effect. So it's 2 bugs, not 1.
That's what I call teamwork.
It is teamwork making the dream work. And then they go and they take and make an E. coli that has that. So they go and look for genes that can do this, and found. So they went and then said, "Hey, can we do this and figure out a way that makes ILA?" So they do find the ILA gene. They shove that into an E. coli, and that pumps out. And if you give that then leads to more IPA in the end. So pushing, Le Chatelier principle, like shoving the precursor levels up high, makes more of the final molecule come in.
Right.
And so then they say, hey, does this work in CRC-derived organoids? In humans, they actually do a air-liquid interface system so you can keep the immune cells alive from the tumor in colon organoids and show that you do have a shift in state. If you just do regular organoids, all the immune cells die during the prep. So they do an air-liquid interface. They show hey, human samples, it works. They also go into other mouse models. And that's why they have this note about pan cancer, is it works. They throw it at a whole bunch of things, which let me get the list, because it's an aggressive list in a good way, right? But melanoma, it works in. They obviously did that one because who wouldn't do melanoma, right? But they go and really prove that this isn't just microsatellites stable. They do breast cancer models. They do melanoma. I guess that's the 2. So that's why they're saying pan cancer, melanoma and breast, but then also it works in people, at least in terms of the explants of explanted colon cancers from people,
right.
Organoid, you can see immune cell activation in the system and killing of the tumor.
That's very interesting.
So there you go. One bug hands a metabolite to another bug, and that bug makes a metabolite that causes acetylation of an important promoter region, which drives immune cell activation. But not everyone has equal amounts of these bugs and thus equal amounts of the metabolites, and so your mileage may vary with your PD-1 inhibitor depending on your gut bacteria, folks.
That was a long sentence. Well, I mean, it has there's already a bunch of studies, right, showing how important the microbiome composition can be to at least PD-1 response. So I think that is fascinating. Yeah, I think we ought to be, colonizing patients with the right bacteria before starting with the therapies.
Yeah, maybe.
Yeah. Is that in the pipeline, Jason? Is that in the pipeline?
Don't ask me questions I can't talk about, Brenda. Definitely it's something people are looking at, generally speaking. I will say that.
All right. Okay. Disclaimer. All right. So to finish today's stories, I have a short story, but about bad microglia. Let's put it that way. The paper is called ApoE4. ApoE4 is linked to damaging lipid droplets in Alzheimer's disease microglia. And first authors Michael Haney and Robert Pavlovics from the lab of Tony Weiss-Correa at Stanford. And in this paper, they studied, trying to understand what are the mechanisms of Alzheimer's disease, and one of the things, and how certain genetic risk factors are associated with the development of the disease. And one of the known risk factors is having this ApoE4 homozygous genotype. So ApoE has, I think 3 or 4 different. I think four is like it's four, but I don't think it's actually four. Um, Different isotypes, and the number four has been associated with increased risk of Alzheimer's. And so what they do is they look into brain tissue of Alzheimer's patients and controls. And one of the things they zero in is they start doing some single nucleus RNA sequencing. And this is fun because this is something that you can do with brain tissue, and it's very commonly done when you're studying this kind of tissue. And they identify, so in the microglia, so it's always the microglia, always immune cells, these brain macrophages. And they identify that based on the transcription, and they see this kind of microbial state, and in which when they compare patients with controls, and in particular they compare ApoE4 patients with controls, there's a couple of enzymes or a couple of genes that really pop up, and one of them is this enzyme called ACLS1, which is it's acyl-CoA synthetase long chain family member one. So it's basically a part of the generation of lipids, of like long chain lipids, and really really kind of pops up from the sequencing data. And this correlates in a way with what they see in the cells, because they do see that lipid droplets, which are associated with disease, are kind of strongly seen in ApoE4 patients and Alzheimer patients. So they study, they really kind of compare patients and the different the ApoE, and compare to ApoE3, and they really see that this lipid accumulation is linked to the Alzheimer's disease pathology. They're really they form these lipid bodies that are prevalent in Alzheimer's disease brain tissue, and they correlate negatively with cognitive performance in the patient they were taking from. And they kind of propose that this accumulation of lipids by the microglia is really inducing and is damaging the neurons, and is inducing phosphorylation of tau and apoptosis in neurons. And they try to find a way of, like, they propose that by preventing the generation of these lipid droplets, they can reduce the severity of disease. So they do some tests in which they find that by inhibition of P1 kinase. They have a like CRISPR knockout screen, trying to find druggable targets that would reduce the generation of lipid droplets, and they find that inhibiting P13 kinase, which is known to be kind of downstream. Lipid production is downstream of this kinase, can reduce lipid droplet accumulation in this this microglia model. And one of the things is like it's associated increasing of autophagy, which also has been suggested as beneficial for preventing or for reducing the severity of Alzheimer's disease. So I guess the conclusion that they take is that this microglia, uh, one of the pathological phenotypes that they acquire in Alzheimer's is this accumulation of lipids, and especially if you have ApoE4 genotype, it's increased, and this accumulation of lipids is neurotoxic in an ApoE dependent manner, and it can really damage the lipids, like the neurons. It can neurons accumulate lipids. They don't express this enzyme themselves, but they suggest that they are accumulating lipids from the microglia, are transferring lipids to the neurons, which is toxic and really reduces their survival and induces part of the pathology of the disease. So it's Yeah, it's kind of sad because the microglia are supposed to be there, to protect the neurons, but it looks like under these conditions, uh, they can be induced to a fairly pathogenic phenotype, and this is actually super detrimental to the neurons they coexist with.
The brain is fragile, clearly. I mean, like, it's interesting, like, too much protein bad, too much lipid bad. Like, just got to be just right.
Yeah, yeah, it really like Goldilocks zone, right?
Mm-hmm.
Yeah, take care of your brain.
Very much so. Well, hopefully we've been filling your brain with everything here, and we're going to be filling it up even more by speaking to
Mhm.
Dr. Scott Canna at the Children's Hospital of Philadelphia in just a moment. But before we do that, the Lab Coats and Life podcast is a new podcast that aims to keep you informed about useful soft skills, relevant trends, and important efforts from within the scientific community to help you thrive as a scientist. Brenda and I are co-hosts for upcoming episodes on the role of open data and science and using social media to make an impact. Find the podcast on your app of choice or learn more at www.labcoatsandlifepodcast.com.
Today, for the second part of our show, we have joining us Dr. Scott Canna. He is Associate Professor of Pediatrics at the University of Pennsylvania and attending physician at the Children's Hospital of Philadelphia. And his lab looks into the intersection of autoinflammatory and hyperinflammatory diseases and how this could be understood to develop new therapies and therapeutic strategies to alleviate this kind of diseases. Thank you so much for joining us for the Immunology Podcast. We are very happy to have you on.
No, it's my privilege. I'm a big fan.
Thank you for being here. All right. Well, I'm going to dive in because one of the things you study is something that was beaten into me, for lack of a nicer way to say it, in residency, which is HLH. Hemophagocytic lymphohistiocytosis, or as we're just going to say for the rest of the podcast, HLH. So I would bet most people haven't heard about that. So I don't know if you want to give an overview and then a little bit about why it's scientifically interesting or what you guys are doing about it in this context of immune activation, because it's a weird one.
Yes. Part of the reason it's weird is because the nomenclature kind of a mess, and I am both responsible for and trying to work against that. And the problem really comes down to, both scientists and physicians use the term HLH to mean two different things. And it creates issues when you conflate one with the other. And so I'll explain. As defined currently, HLH is not really an etiologic diagnosis. It's kind of a description. And the way that I try to teach it is like there's a descriptive diagnosis called shock that we use all the time, which not to say HLH is shock, but just that we understand that shock is sort of this physiologic state, and there's a lot of different things that can go into it, right? You can be infected and have, distributive shock, you can have cardiogenic shock, and you can have multiple at the same time. But the physiology of shock is a thing that we recognize and is worth recognizing in and of itself, in addition to it sort of triggering your brain to think about certain things. And so HLH is in a little bit, in a sort of overly simplistic way, kind of like T-cell shock. If your T cells are really massively activated, you're going to get this immunophysiologic entity that we call HLH. And that consists of the things you might expect, really high fevers, drops in all the different cell counts, anemia, thrombocytopenia, this sort of microclots all over the place called disseminated intravascular coagulation, or DIC. People can get hepatitis, they can get big spleens. The biomarker that's probably best known is this one called ferritin, which can get just astronomically high. And then for whatever reason, the damage of HLH really seems to like the liver and the brain, for reasons that we're still working on, but not quite sure. And so, writ large, when we say HLH, we kind of mean this immunophysiologic spectrum that can be associated with infection or cancer or certain treatments, like basically CAR T cell cytokine release syndrome and sort of the second line of that are all kind of within this umbrella. But sort of mechanistically, when people say HLH, they're usually referring to a really cool sort of mechanistic problem of impaired granule-mediated cytotoxicity. So if you've got NK cells and CD8 T cells, and they can, develop normally, and they can get antigen presentation normally and make an immune synapse, but they lack perforin, or they lack a bunch of the machinery necessary to get those perforin-containing granules to the lytic synapse, they never really shut down that immune synapse. They never sort of shut down that very stimulatory kiss, and it proceeds for far too long. They pump out a ton of interferon gamma and other cytokines, and you get that phenotype. And so that's been incredibly instructive. The confusion sometimes comes if you treat everybody that has that sort of clinical immunophysiologic state like they have a granule defect, you're going to treat people wrong. And so I will sometimes glibly say, like, do you mean HLH or do you mean HLH? And the proper term for the granule mediated thing is familial HLH. But it's often, hard to remember to use the most precise language.
So there's kind of this one that is associated with a defect in your immune system in which you inherit an incapacity to produce these perforins or kind of give closure to these cells. And then there is the acute version that you see when, for example, you have CAR T cells. So I didn't quite understand. Is that also HLH, or is it only the ones that are associated with more of a genetic defect?
So HLH, the immunophysiologic state, covered lots of different things, including familial HLH, which is the genetic defect in granule-mediated cytotoxicity. It turns out there's lots of other contributors to that phenotype. And I'm a rheumatologist by training. And so, the one that's closer to us goes by yet another name called macrophage activation syndrome, which is another sort of subtype of what we call secondary HLH. I know it's a mess, I'm sorry. But within rheumatic disease, my lab's really been focused on IL-18 because it really seems to be both kind of an extraordinarily specific biomarker and probably upstream of damage in patients that develop this other subtype. And for those patients, their cytotoxic killing is, as far as we can tell, completely intact. In fact, maybe even a little better.
I guess now that we're discussing semantics, I think there's maybe a word that it seems to be a key word in your research, which is the concept of hyperinflammation. What exactly? Is just a big inflammation, or what do we understand exactly as hyperinflammation? That correlate with, we mentioned IL-18 and what comes after or kind of beyond IL-18.
So, I think hyperinflammation and the broad definition of HLH are kind of pretty much completely overlapping Venn diagrams. Some people would sort of throw in cytokine storm into that, but I think with the pandemic, cytokine storm kind of got popular and it was everywhere. And so if it's everywhere, it's kind of nowhere. But yeah, another way that I think of hyperinflammation, and this is more just sort of a mental exercise for the way that I think of mechanisms of immune dysregulation, is kind of a excessively massive response to usually a pathogen antigen. So it's not autoimmunity because it's not usually a self-antigen. It's not autoinflammation because it's still antigen specific, but it's still a much exaggerated response to what would otherwise be a normal, antigen mediated process.
So is part of the confusion that, if I remember right, HLH was originally a histology finding, like someone would come on with symptoms and they would see something very specific for HLH, which always was a board question, or at least, a grand rounds question. Is that part of the etiology of the nomenclature
Yeah, I think that you're absolutely right. You can't really divorce yourself if you work in biomedicine in any way. You can't really divorce yourself from like the history of medicine. And a lot of the names that we use and a lot of the groupings that we use come from when, looking under the microscope was about as much as you were going to get. And so there's these kind of very cool looking activated macrophages. The old timey term for an activated macrophage is a histiocyte. And so if you see an activated macrophage in the blood or in the bone marrow that has a bunch of other cells inside of it, red blood cells, neutrophils, lymphocytes, you call that a hemophagocyte. And so if you see a whole bunch of hemophagocytes, that goes along with HLH. It turns out if you look for them, it's neither sensitive nor specific. So it's kind of cool, but from a diagnostic standpoint, not so useful.
But that's where it came from.
That's the hemophagocytic lymphohistiocytosis. And it was lumped in with a whole bunch of other histiocytic disorders like Langerhans cell histiocytosis. And, there's a bunch of Rosai-Dorfman. There's a bunch of rare ones that now that we understand the mechanisms a little bit better, we actually think, for pretty much every sub-flavor of HLH that it's kind of actually a T cell problem, and that the macrophages are kind of taken hold of by this sort of overwhelming T cell response.
You see, Jason, it's always the T cells.
Yeah, troublemakers.
But sorry. So you mentioned if we may get a little bit onto the details, because I don't think we spoke ever about IL-18 on this podcast. So I think you seem like the right person I know to ask. So what how does IL-18 work how does it contribute to inflammation or to hyperinflammation? And within the work of your lab, of your group, what kind of what have you found and in relation to these diseases that you study in patients and particularly in children?
Yeah, I mean, we are definitely in love with IL-18 in my lab. So it's a super cool cytokine for a variety of reasons. We just had a nice review in Nature Reviews Rheumatology that goes into the excessive depth, but it was originally described among like this group of what were called interferon gamma inducing factors, and within that group of proteins included IL-12, P70, and IL-18, and as they sort of were able to clone out the different proteins that were interferon gamma inducing factors, IL-18 was one of them. And so it's really good at inducing interferon gamma. Its production is a little bit different. It's an inflammasome activated cytokine. It's kind of there at really high levels as an alarmin, probably in pretty much every barrier epithelial cell, skin, intestinal epithelial cells, bronchial epithelium, at pretty high levels. But it's inducible within macrophages in kind of strange and mysterious ways that we don't really understand. It's not inducible the same way that other inflammatory cytokines like IL-1, IL-6, TNF are. So, it's in those spots. It's got to have usually an inflammasome to get cleaved and activated. But then once it's active, it's still got to get outside the cell. And so it requires usually Gesderm D pores, also part of the inflammasome, to get out. And then once it's out, it kind of gets immediately neutralized by this soluble inhibitor called IL-18 binding protein, which has a really high affinity for IL-18, pretty much irreversibly binds. And so in order for IL-18 to signal, it's either got to signal kind of in a autoparacrine way, or you got to be making so much that you overwhelm this sort of IL-18 binding protein reservoir. And then the receptors are on the cells that we tend to think of as sort of gamma producers. So activated CD4s and CD8s, NKs, ILC1s, and that kind of stuff. And you can find the receptor on other things like ILC2s. You can find the receptor on some granulocytes, actually, where we're not quite sure what it's doing. But the way that we think it's predisposing people to this subtype of HLH that for historical reasons we call macrophage activation syndrome, or MAS, is probably through amplification of T cell responses, which it's really good at, in particular type 1 Th1 responses. And that, those an otherwise typical T cell response just gets massively amplified. And then, gamma itself is kind of a funny cytokine and likes to propagate its own inflammation. And so it can be a little hard to turn off an interferon gamma driven sort of cytokine storm, because interferon gamma and other interferons are pretty good at sort of cutting off some of the switches that regulate themselves in the way that, other like TNF and IL-1, those kinds of things exhibit tolerance pretty quickly. And interferons are pretty good at subverting that. So we think IL-18 is mostly promoting those amplified responses. From a biomarker standpoint, like I'm also a clinician, we measure inflammatory biomarkers and cytokine panels all the time. And almost everything is staggeringly nonspecific. IL-18 at really high levels is pretty amazingly specific for like a small group of autoinflammatory disorders that tend to predispose to this MAS thing.
So I want to go back to the soluble inhibitor because it's cool. Is that ever regulated? Like, do its levels get tamped down as part of a pro IL-18 environment? Or is it really, it's kind of steady state and you just got to overwhelm the sucker?
So at baseline, humans and mice make a lot, but it's an interferon gamma response protein actually. So it's probably part of a feedback loop on 18. And so, the cells that can respond to interferon gamma are good at making binding protein, which is pretty much every cell. And so, the levels certainly do go up. It's actually a pretty decent interferon gamma activity biomarker, but we tend to use CXCL9 for that clinically. So it's around somewhat constitutively, but the levels go up in response to gamma.
Maybe switching a little bit and focusing a bit more on your clinical work. I think one of probably the scariest diagnoses that a person can get is your disease is idiopathic. We don't know what's wrong with you. You have, and I think that happens. So it happens a bunch, especially in this inflammatory diseases, autoimmune diseases. And I know that you are involved in and trying to understand and trying to systematically look into these disorders in children and trying to find answers to those that don't know what's going on. So would you like to talk to us a little bit about what happens when you see these disorders in the clinic? What is the most common things that you see, and how do you go about trying to understand and provide patients with an answer?
No, great questions. And has sort of been like the preoccupation of my entire career since about like 2005. Because, every new or ultra rare disease was was idiopathic before somebody figured out what the gene was. And so, patients with these diseases have been around forever, but our ability to detect them really evolves over time. Obviously the whole revolution in sequencing by synthesis or next gen sequencing, if you're an old timer, has made it a lot easier to detect some of these variants, and so we're moving into sort of next level evaluation of those things. But it used to be, that you had to join some sort of research study, often at the intramural NIH, where I've done some training over time, and they've discovered a bunch of diseases. But even then, most of the patients they would see would kind of go undiscovered, undifferentiated. And so I work at the Children's Hospital of Philadelphia, and round about 2013-ish, largely in response to these really complex and very sick HLH patients, a group of the doctors here put together a HLH response team, which was really just a group of interested individuals from a variety of different divisions. There were hematologists and oncologists and infectious disease docs and rheumatologists and PICU people who at least agreed to talk with each other in a constructive way when these confusing cases would come up, in a way that would be, most clinically efficient, minimize yelling over the bedside, and hopefully get to the right diagnosis, or if you're not going to get to a precise diagnosis, get to the right ballpark as quickly as possible, and be creative and appropriate about treatment. And so, that worked and has evolved over time, to sort of expand into what we call our immune dysregulation program. Internally, we call it the Dysregulated Immunity Response Team, or DIRT, but that's not I guess that's not something that marketing thought was going to go on the website. So, it's a multidisciplinary program. It's both inpatient and outpatient. And it's really been, I think, just an amazing resource of, interested clinicians who, almost all of us, run individual research programs that have a sort of tie-in to the kinds of patients that we see within our immune dysregulation program. If I slip and say DIRT, you'll know what I mean. It's an amazing training opportunity. It's an amazing learning opportunity. And it's an amazing scaffold to do the kind of natural history research, disease discovery or biomarker discovery, validation, that kind of stuff. So the kind of diseases we see, we still deal a lot with HLH, both on the inpatient and outpatient side. But really, anytime that there's things that don't quite fit or make sense, whether there's sort of multiple weird autoimmunities, or autoimmunity in someone that also maybe has a cancer susceptibility background, or combinations of immunodeficiencies and autoimmunities, really wherever we can be helpful, which turns out to be a lot of places, because as you can imagine, patients can be kind of complicated and pretty sick. I'm a rheumatologist again, so even when we have like a pretty straightforward scenario, we still don't necessarily know what causes the disease. Like, lupus is still kind of a black box for a lot of patients. It probably represents a lot of different diseases, and that's supposed to be sort of one of our bread and butter. So the immune dysregulation program is a multidisciplinary program, probably among, if not the biggest in the country so far, to try and approach these situations in a more standardized way, and in a way that interfaces with the state of the art, both from a diagnostic and treatment standpoint, but also from a research standpoint.
So along these lines, with a group like this, you can make better diagnoses than, hey, it's this HLH thing because I see some weird cells, because you said that wasn't very useful. And now next gen sequencing and all this comes along the line. So I'm going to ask the clinical follow-up question. What can you guys do with that information to help patients therapeutically?
Oh, gosh, I would say quite a lot. I mean, without getting into the sort of deep weeds of like, oh, how do we, stage this workup, and when do we send genetics versus, like everything, it's an it depends.
Take it a step back though. Like, are there drugs that can blockade IL-18 specifically, or steroids, or you just like, I put everyone on Tacro and walk away anyway, to which then I would say, which I know is not the answer, but to play that out, then I'd go, it doesn't matter what it is.
So it's super, it's super complicated, which makes it kind of really fun and interesting. There's plenty of patients that you wouldn't want to just give them steroids right away, because you haven't ruled out whether they have a cancer. And giving steroids to somebody that might have a lymphoma is going to make it real hard to find that lymphoma. And eventually when it does come back, it might be a lot harder to treat. So, there's lots of individual cytokine blockers. We use this IL-1 blocker called anakinra a good bit. There's an FDA-approved interferon gamma blocker that we use a bit. There's a whole slew of these specific kinase inhibitors, like JAK inhibitors, that we use more and more. And there are some, investigational things. There's no FDA-approved IL-18 blocker, but we have, we've had a couple clinical trials running, and I think we have 5 or 6 patients on extended use, compassionate use treatment with these things. So yeah, the program is helpful for trying to get multiple kind of orthogonal opinions about what the right thing to do, and then, getting access and executing some of these things that are quite, non-standard, whether it's in compassionate use setup, or whether it's something that's, quite off-label because these are little kids and nothing's on label. Um, there are a slew of options, but they're all double-edged swords, and so making good decisions is not always easy, and often multidisciplinary keeps you from making mistakes.
So could you talk about the challenge a little bit of treating kids? Because as you said, nothing's on label. And you're like, well, wait a minute, there's all these drugs out there, but being approved for adult and being approved for a child are not the same in the US. So I don't know if you could speak to that a little bit, because I think it's something CHOP is really good at navigating, but is also like a problem in therapies. And it makes sense. You don't want to give kids things necessarily, but who's going to do a trial on little Timmy?
Right.
But like, how do you guys navigate that, and any thoughts on that space for clinical therapy and what we should be doing better?
I mean, yeah, it's a big problem. The FDA has done a little bit to require some, at least sort of phase 1 pediatric plan when you're developing something. But, like you said, it's rarely a moneymaker to get a purely pediatric indication. And so, the off-label use of drugs is kind of the rule for a lot of even our bread and butter diseases, because there just aren't studies in kids. And so, we have an obligation to do things that are tested and safe, but we also have an obligation to do the best we can with the tools that are available. And, if a drug is FDA approved for something, you can use it, carefully for something else. And like I said, for some pediatric diseases, even the ones that we don't consider that rare, there's like pretty much nothing that's FDA approved. So there's a lot of sort of extrapolating from experience, or from the kinds of clinical data that wouldn't result in an FDA registration. But as you can imagine, like getting an FDA registration is pretty expensive. And so, as part of a variety of research networks that I interface with, there's a variety of different plans for how do you get a drug approved for rare disease. Not just how do you accumulate a little bit of data to show that it works, but how do you structure the data acquisition in a way that is going to result in, this getting an FDA indication, and then who's going to pay for that? And I would say that there are some good ideas, a little bit of execution in that area, but still a lot to learn, both on the side of the researchers and definitely on the side of the FDA.
So you basically have all the complications together. You have rare diseases, children, and unauthorized drugs that you need to kind of justify using them technically off label. And then you're trying to make the most out of that situation. Must be very tricky.
Yeah. I mean, it's never not interesting.
Yeah.
I wouldn't say that pharmacy benefit manager companies have been helpful.
Yeah, let's not get into that, because then we would end on a pretty somber note. I just before we move subjects, I just want to make a suggestion for the name of the program. If dirt is too complicated, you could go for dysregulated immunologist program, DIP.
Yeah,
feel free to take it if you want.
the immunologists are dysregulated cognitively for sure.
So, aren't we all? But it's been very interesting. So I think maybe we can start wrapping up our conversation. I learned a lot, and I think it's very important to have these initiatives trying to systematically provide answers, and like pulling together all the knowledge and all the strategies. I think that must be complicated, but I'm sure is going to be very useful in the long term. So at the end of our talks, we like to ask our guests some questions to get them to know better a bit, and to our audience to see them beyond the lab. So as our fun question for you today, we would like to ask you, what is the biggest misconception about science that you would like to correct, and why?
Oh man. So yeah, not to get like overly philosophical, but I think again, there's sort of this conflating of words. So I think of science as a process um, of getting towards what is agreed to be sort of the best consensus of objective reality, which means it's going to change all the time in in response to new evidence, new data, new thoughts, and like that's all a good thing. But I think that we often refer to science almost like we want to say, oh, the best science, or what the science says. Um, not referring to it as a process, but referring to it as like the state of the art.
Mhm.
And I think it's worth sort of, I don't know, thinking about them both somewhat separately, with the understanding, because we get ourselves into trouble, right? We say
Yeah.
like, oh, well, science said my family gets a lot of food allergies. I shouldn't expose my kid to peanut butter till they're 5. And then, well, then science was wrong. Now it says to give it to them when they're babies. And like, well, yeah, that's how this works. We were wrong, and we did some studies, and the science has changed. So I think that, like, if we think of science as a process, it allows for iteratively better refinements of what the state of the art is, and it also kind of reinforces the fact that like what we're shooting towards is like objective reality. And so,
Mm.
when you disagree with the science without contributing to the data or the understanding or the analysis, you're basically disagreeing with objective reality in a way that is, often not just dangerous to yourself, but dangerous to like the entire state of Florida, for instance.
Less somber than pediatric drugs, but still a philosophical question. But yeah, I think you have a point, right? There's science, the process, which produces science, the current body of language, and the process constantly proves ourselves to be idiots.
Right.
Which is fine.
Which is fine, but people don't like that.
No. Well, I mean, not to make everything about COVID, but I think if anything has shown at a larger scale how important that concept is our ever-growing understanding of how the pandemic went, right? I think that's.
But like,
I don't want to.
I don't know. I mean, I don't. I'm not a vaccine researcher, but like, obviously, I'm a pediatrician and I'm biased. But like, I'm like a vaccine evangelist, and we got real lucky with the rollout of a bunch of incredibly effective, incredibly safe vaccines that, like, kind of saved the world a bit. And so I don't know that we rah-rah that enough. And part of the reason we don't celebrate that quite enough is maybe because there's a bunch of companies that made those vaccines that, have profited handsomely from doing that. And, we can argue about how much they should profit from vaccine development, whatever. But, at least we got a lot of really good vaccines. And that the knowledge that we learned from that, I mean, look at the RSV trials, right? Like so I think the propagation of these things that contribute to the body of knowledge that we call science is something that, like, we should spend some time celebrating.
Well, on that uplifting note, definitely we should spend time celebrating the how would be the positives or the accomplishments. I don't think we do that enough. I don't think we share that enough, especially with the general public. Um, and I think I think always the conclusion is we should do it more. So,
all right. Well, with that, thank you for coming on.
Anytime. It's been a pleasure.
yeah. So thank you so much, Scott Canna from the University of Pennsylvania and the Children's Hospital of Philadelphia, joining us today. That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.